So we'll move right into the panel discussion. So if I can have the last four speakers, please come up to the front table and we'll start taking some questions. I will try to lead in with a couple of questions that were not actually brought up in the discussion, and I'll open it up to the floor for a couple of questions as well. So one of the questions that was submitted ahead of time is, how successful are academic drug discovery programs compared to big pharma? So who would like to take a stab at that? Go ahead.
I don't know if success rate's the right way to measure it, but I think it's a matter of mindset. And I think if you look at for some companies, the percentage of their pipeline that was not developed internally is now approaching 60–70%. So I think what's been learned is that industrializing this process can be very challenging, and that sometimes the scrappy, hardworking, entrepreneurial approach, where you're much more willing to kill a bad idea sooner, may at the end of the day be more cost effective.
I think, as Greg has just said, about 60% of marketed drugs came from academic labs originally, so overall pretty successful.
Absolutely. Yeah, thank you very much. Okay, another question that was submitted is, what are the distinctive issues that differentiate academic from commercial drug discovery? So go ahead, Michelle.
May I? I'll hit the button first.
This is not a game show. Go ahead.
We're not going to be competitive about it. It's just so much more fun if it's competitive. No, just kidding. Our focus is on innovation, right? Like we talked about, you don't want to develop the next statin. Well, the next statin might be an extremely valuable commercial product, and the sixth statin was way better than the first five, but that's not what academics are going to do. What academics are going to do is the first OLIG-2 antagonist, or the first transcription factor in glioblastoma. So the first things to market is where academics, I think, should be focused.
Very good. Okay, this question I think will go to either Phil and or Michelle here. So there were multiple questions submitted on DNA-encoded library screenings, which I know... Okay, sorry. Sorry about that. There we go. So I think this question will be directed at Michelle and Phil probably. They didn't get really a chance to touch on this in their presentation, but several people that have joined us here in the audience today are curious about the DNA-encoded library screening and the future of that, and what their perspective is on using that technology for drug discovery purposes.
So I think, as Michelle said in her talk, there are multiple ways to seed a program, and DNA-encoded libraries is a technology there, and it's a very valuable technology, and there's multiple ways to get leads.
Yeah, we're just starting, so we submitted a bunch of projects. We have a collaboration with a big pharma that has an expertise in DNA-encoded libraries, and we haven't gotten the data back yet, so next time I'm invited to talk, I'll give you an informed opinion. But what I'm really attracted to about it is that you can, because it's a pooled assay, you can use a lot of controls. So you can get the molecule binding to your target, but not to a mutant, or not to the target with the substrate bound if you want a competitive inhibitor. So you have a lot of room to play. So it's seems like a hypothesis-driven kind of screening approach but I don't have any data.
And just like any other screening-based effort, I mean, that's on a large scale. You do need to be patient. It isn't something where you'll have data overnight. You can certainly assay a greater diversity of chemical matter than you can in a traditional high-throughput screen, but it still takes time. You need to have patience for that as well.
Yeah, I think, sorry, just the other point about that's a good point, that you have to do chemistry faster. Fragment-based drug discovery, also, the screens are very fast to do, but you need structural biology and chemistry really early in the process. So one thing that's attractive about drug repurposing or traditional high-throughput screening or cell-based high-throughput screening in academics is that the chemical matter has a higher hurdle to get discovered, and so you can do more with less chemistry in the early stages.
Great. So there's one question in particular I thought was pretty profound here, and that is with respect to what are the emerging trends in drug discovery and development. So I think all of you could certainly contribute to that particular discussion. Should we start on the left and work our way towards the right, Gregory?
Well, I'm down on the ground, so I don't have the 30,000-foot view, so I'm probably not the best person to answer that question.
So I'll take a stab at that one. So I think there's two areas that is changing. One is the breadth of modalities out there. I mean, before you used to think about kind of, if you were lucky, it was a peptide or an antibody. Today, it can be anything. It could be, we see antisense oligonucleotides approved. We're seeing cellular therapies. We are going to see vaccines out there as treatments. So that's evolving. But I think the other ways that you're seeing is moving into other diseases. I mean, there are a lot of companies being founded now targeting NASH. There are companies out there for, about hearing loss. So I think kind of we're moving into kind of medical need that hasn't otherwise been addressed.
Yeah, I agree with those. The different modalities, that's a really profound change. And also, so even in the small molecular, the more traditional modality space, we see much more quantitative and sophisticated biology moving closer and closer, earlier and earlier in the process. And that's what's making some of these things accessible. And also, structural biology is advanced to where we can look at some of these undruggable targets again in a new way. So we have the advancements in biology, the advancements in high-performance computing that's going to have a big impact on how we computationally predict what molecules are going to do, and new structural approaches. And together, I think that's just saying, okay, what's the important biology, what's the important way to handle it, and a much more integrated view of what can be done.
Exactly. Okay, very good. So, yes, please go ahead.
Hi there. When you're developing a drug and moving into the market, the federal agencies are now looking at new training programs for trainees, graduate students, and fellows. So could any of you speak on the I-Corps program and NIH and NSF, the Cancer Institute, moving into the need of that training for individuals that are going to commercialize? So does anybody there have any information on I-Corps that they could bring forward? Because we've got 4 centers that train here in Houston.
I can't speak directly to I-Corps, although I'm familiar with it. What we are seeing, though, is more and more it's becoming obvious that we need to train scientists to better understand the business. And what I can speak to is a group we founded at Johns Hopkins called the Society of International Bioentrepreneurship Education and Research, CYBER, which is really a consortium very similar to this, except that it's global. So educators from all over the world come together to talk about what do we need to train people to do. So the federal government, NIH, et cetera, are also seeing this desperate need.
So here in Houston, the University of Houston has an I-Corps program, Rice University has an I-Corps program, and so does MD Anderson. So that may be something that maybe you might want to include, for the materials.
So having actually spoken to a couple of the I-Corps trainees last week, and then actually knowing some of the people that run the program last year, I mean, I think it's a fantastic training program, and it's a great way for people to kind of test drive whether their science, whether it will fit for commercialization. So great to hear that there's multiple avenues here.
No, please. Are there any other questions? Just go ahead.
Yes, I'm Antonio. I'm BD Medical Metrics. And my question is, at what point do you start to have conversations about patient selectivity early on in the development process for your drug design? And also, after the drug is already in the market and there is some issues with toxicity, do you go back into your books and start to revamp your design process, perhaps address those?
So if I understood the question, at what point do you start talking to the pharma companies and the patients about?
Right, so when do you start to have discussions about selecting the right patient population to run your trials? And even early, as was mentioned, early on in the development, the design process of your drugs?
I think as soon as possible. I mean, when I was starting my current company, I was going into an indication, glioblastoma or GBM, that it's by many sort of considered a third rail, and so the first question I had, and I went out very early, I brought in a BD guy, and we went out and talked to a lot of the big pharma, is this even an area you would be willing to invest in? And if so, where along the process would you potentially jump in? And importantly, what's the data set going to need to look like? I didn't want to make assumptions about what that data set was going to need to look like. I wanted to hear from a variety of people what their hurdles were and what their check boxes were, so that we could then go back and design our critical path drug development program to fit that objective. So as soon as you can. I don't think there's any harm in going out and talking to patients, patient advocacy groups, multiple stakeholders, to get as much information as possible.
So one of the things that hasn't come up thus far is kind of also about how you're going to sell your product. Are you going to go there with a product and an in vitro diagnostic? Because that brings its own challenge. If you're going to sell your product only for a subset of patients, you have to develop both things simultaneously, and it's fundamental that you kind of invest earlier, as Greg was just saying.
Thank you for your presentations. They were very insightful. My question is regarding artificial intelligence, and how do you think it has been implemented into the research process? If you still think that there are areas of opportunity, or it has been fully implemented?
So I'm part of this the ATOM consortium that Mary referred to as it's AI-driven consortium, but the idea is that bringing together all that data that's in pharmaceutical companies' database that isn't really well integrated into the next drug discovery project, except in the head of the medicinal chemist, usually. So how to take all of that data, put it into a system where machines can make use of it, because it's so large, diverse, most of it's negative data, right, didn't hit. And these complex in vitro models, human relevant models, and supercomputing. So we're working with the national laboratories, with Lawrence Livermore and 3 other national laboratories and pharmaceutical companies, to try to do what you're saying, try to fully implement it. What I've seen now, this is definitely not my full area of expertise, but what I have seen is a number of companies that are very mature at a piece of that pipeline. So really good at PK prediction, or at docking and modeling, computational modeling, or pharmacophore prediction. So these are the early stages of binding and efficacy. But taking those same models and integrating them, and thinking about structural models and safety. Because you're binding to a target, right? We think of safety as the animal died or the liver got purple or something, but it's molecules, it's proteins that it's binding to that are doing that. So AI has the ability to work at all these things in parallel eventually, and I'm sure it's in style right now, and there'll be a lot of pitfalls that we don't yet know, but I think it's going to have a big impact.
And I think going forward, we're still going to. I don't think it's found its home today, but it will find its home with time. So, looking out into the auditorium here, everybody's a lot younger than I am. People should be paying attention to it and understanding how it can impact in their science as they move forward.
Any other? No, please go ahead.
I have a question about kind of the mistranslation between animals, preclinical and clinical studies. So when you get a molecule in preclinical studies and they don't translate well in the clinical studies, is this down to, so you also talked about selecting the appropriate animal model. So do you ever foresee in the near future being able to move molecules to the clinic without animal models?
No.
Oh, you can do that now. You can do that now.
Oh.
Yeah. So the cystic fibrosis drugs don't go through animal models. Animal models are no good. So use patient-derived lung epithelia, measure ion channel trafficking and function, mucus movement or something, whatever the key.
Yeah, there are
So
organoids being developed, and
Right.
some systems, but it's going to be tough, and in general probably not.
For efficacy, right? So it's efficacy, it's really up to you. It's your own comfort level and your investors and what you're willing to do. And there is a lot of effort to make animal models better. But also, I think what Greg's saying, complex in vitro models and looking at more complex cell cultures and primary human cells, disease cells, is, I think, going to have a huge impact. The big hurdle that's going to be harder to cross is getting away from animals for toxicology, but I think is similarly going to have its breakthrough eventually.
Go ahead.
First is addressing the ICO. I think many years we started like 4 years ago. I think I was among the first group who started doing that. I think I talked to several people. They did the ICO program like last week. I think for the drug discovery and development, it's very difficult for me. I mean, I also talk to them, they agree with that because they ask you to go out of the building to identify customers. But for drug discovery group, I think it's kind of hard to identify a customer. I mean, our end user will be the patients, right? But they don't really pay the check. I mean, write you the check at the beginning when you started the program. I think for us, usually it's like identify the customer would be the executive from big farmers or venture capitalists or angels, but particularly for West Coast and East Coast, maybe it's a little bit easier because you have a lot of company there. For Houston area, I mean, we are struggling to do that, but it has been, still, we are still doing that. So I think that's the challenge I have experienced, and a lot of the ICO programmers experienced, but we are doing that. So for the specific question I have here is that, I mean, I know a lot of people, you talk about the drug discovery development from the business point of view. I mean, you have the fail early, fail fast, and you have to have the endpoint and try to have a go, no-go decision. But for me, because we are doing, innovation, we are doing research, right? I mean, for example, the Gleevec, so initially it was designed for PKC inhibitors, but later on it was BCR-ABL inhibitors. And then in Viagra, first because of toxicity field and then become a blockbuster. So, I mean, from the academic point of view, or research point of view, what do you think of this kind of fail early, fail fast, or the go or no-go decisions? I mean, how clear you want to be to do that kind of things?
So, I mean, you always start with a hypothesis. If you test that hypothesis and it works, you move ahead. If you test that hypothesis and it fails, you typically stop or you park a program. Just because a program has parked doesn't mean it can raise its head in a different indication. I mean, if you think a case of Viagra, it was developed for one indication. There was a safety signal, and then it got repositioned on the basis of that safety signal. And so it's opportunistic. You may no longer be pursuing a particular program, but biology moves on and research moves forward in other areas, and occasionally you need to check in and can what you've parked be reinvigorated?
And I don't think it's black and white, but it depends on what your resources are, what your timelines are. But I think you don't serve yourself well if you are not continually asking, what's the killer experiment here? What's the one big question that's really going to answer or get you to that point where you say, okay, this makes sense or it doesn't make sense to move forward, because all the rest is just churning. And you may be answering some interesting questions, but they may not be the important question.
The one thing that I will say as a caveat to the fail fast is not testing your hypothesis because the experiment was incomplete. We have this, we say, oh, well, we ran it through a high throughput screen, we didn't get a good compound, therefore we failed fast. Well, you just failed. You didn't answer your hypothesis, which was, so we favor, if you care about your biology, test as many different approaches as you can to try to get good chemical matter, and then let the chemical biology give you some insight about whether, that's a good tool to answer the critical question. And I totally agree that biology is changing all the time. And we have a project that it's really a target in search of an indication. They keep popping up NASH now as the new indication for this target. So, yeah, just keep a heads up.
We'll have one last question.
This is Lee from Baylor College of Medicine. So I do both basic research and translational study. My question is, I have all the hypotheses tested in animal study. Now I have a good lead for human study. And there are companies show interest to ask me to do toxicity study and what is it called, first in toxicity and other translational study. My question is, in such kind of situation, usually how much money the company can support? And they prefer this kind of experiment to be done in academic institute, or they prefer to do it, to have the drug lead, but do it in their own company.
So yeah, they're talking about GLP, like good laboratory practices, toxicology. So really what you need to go into humans is a highly regulated process. Is that what they're asking for? Yeah. I don't know, a couple $1 million? A lot.
Yeah, easily. And you really need to do discovery safety first, do non-GLP tox, and there's lots of in vitro work too, because there's so many different places that you can get tripped up, right? You can get a herd signal, you can get CYP inhibition, mutagenicity. I mean, there's lots of different assays that you need to run that molecule through, and that, that may send you back to your optimization process.
Yeah, preclinical just means non-in-human, so it's a pretty vague term. I would say that you should try to get them to pay for it. The problem is the more data you have, the more data they'll say, okay, and now this one, and now this one, and now this.
But it sounds like you need a conversation with your tech transfer office to understand exactly what you're holding, what do they want. If they want to talk to you, that you've probably got something valuable.
Yeah, talk to consultants. It sounds like this is a good opportunity to meet with a consultant who focuses on safety and toxicology.
Thank you so much, everybody, for a very interactive session number 1.